Good morning. Today, I'll be discussing about the cementum periodontal complex. So what is a periodontium? The periodontium is a connective tissue that attaches the teeth to the bones of the jaws and provides support of the teeth during function. So periodontium, it includes cementum, periodontal ligament, alveolar bone, and that part of the gingiva facing the tooth. So I have prepared the presentation based on these questions. And now let's start with cementum. So cementum is a mineralized dental tissue covering the anatomic roots of human teeth. It begins at the cervical portion of the tooth at the cemento-enamel junction and continues to the apex. It is avascular and non-innervated. It doesn't have any blood supply, doesn't have any nerve supply. It is continuous with the periodontal ligament on the outer side, and it is firmly adherent to the dentin on its inner side or deeper side. So, classification of cement. There are three different ways where we can classify cementum. The first is based on the presence or absence of cells, that is a cementocyte. And second is by the nature and origin of organic matrix. And third is the combination of these two. So based on the presence or absence of cementocytes, the cementum can be classified as cellular cementum and acellular cementum. And based on the nature and origin of organic matrix, extrinsic fiber cementum, intrinsic fiber cementum, mixed fiber cementum. And the combined classification includes acellular extrinsic fiber cementum. It's also known as the primary cementum, cellular intrinsic fiber cementum, also known as the secondary cementum, mixed fiber cementum, and acellular afibrillar cementum. So, to understand the classification better, you should know the process of cementum formation, or otherwise cementogenesis. So as we all know, the development of the roots begins after the enamel and dentin formation has reached the future cemento-enamel junction. This portion. Here. So the enamel organ plays an important role in the root development by forming Herdwick's epithelial root sheath. So what is Herdwick's epithelial root sheath? The inner and the outer enamel epithelium at the cemento-enamel junction proliferates downwards to form a layer known as Herdwick's epithelial root sheath. And so what is the main function of this epithelial root sheath? This Herdwick's epithelial root sheath induces the cells of the dental papilla to differentiate into odontoblasts. So usually in the coronal dentin, what happens is as the odontoblasts start laying down the dentinal matrix, the odontoblasts move pulpward and it leaves behind the odontoblastic process. But unlike coronal dentin, radicular dentin, as the odontoblast starts secreting the organic matrix in the radicular dentin, what happens is that as the odontoblastic cells move pulpward, they do not leave behind the odontoblastic process in these first few layers of dentin, So, this layer is structureless and it looks like glass. It has got a glassy appearance. So this layer is known as hyaline layer of Hopewell-Smith. So the first few layers of radicular dentin is structureless. The reason is, as the odontoblasts retreat inward, they do not leave behind the odontoblastic process. Now, and that is around approximately 10 micrometer thickness. Now, as the first few layers of radicular dentin is formed, what happens in the Herdwick's epithelial root sheath? Breaks occur in between the Herdwick's epithelial root sheath. And as a result, this newly formed radicular dentin gets exposed to the cells of the dental follicle. And as the dental follicular cells get exposed to the radicular dentin, what happens is that this induces the cells of the dental follicle to differentiate to form cementoblast and also fibroblast. So at this stage, I would like to explain about the difference between extrinsic fibers and intrinsic fibers. As we move ahead, this term is very often comes. So what do you mean by extrinsic and what do you mean by intrinsic fibers? Extrinsic means it is something outside and intrinsic means something inside. So extrinsic means usually the fibers of the cementum, if it is formed by cementoblast, that is within the cementoblast, if it is secreting the collagen fibers, then they are called as intrinsic fibers. And if the collagen fibers are formed by the fibroblast of the periodontal ligament, then we call it as extrinsic fibers. So the orientation or the direction in which these fibers runs, it varies, it differs. So extrinsic fibers, they run in the same direction as that of the principal fibers of the periodontal ligament, that is perpendicular oblique to the root surface. Whereas intrinsic fibers, they run parallel to the root surface at right angles. Okay, so the whole cementogenesis can be divided into two phases. First is the formation of the primary acellular cementum, and the second phase is the formation of the secondary cellular cementum. So like any other tissues like enamel and dentin, here also cementum is first, the organic matrix is laid down, and this organic matrix is later mineralized to form the end product, that is the mineralized cementum. So the first formed cementum is known as the primary cementum. And this primary cementum, it is present at the cervical 1/3 of the root. So as we have discussed about the hyaline layer of the radicular dentin, so the cementoblast that lie very close to the hyaline layer, it synthesize collagen and they form a strong union. At the superficial surface, these collagen fibrils produced by the cementoblast form a fibrous fringe perpendicular to the periodontal space. So the cementoblast, they start secreting collagen fibers onto the hyaline layer and they forms a strong union with the radicular dentin on the inside, and at the outside what happens is that it forms a fibrous fringe on the outside. So the mineralization of the first formed dentin of the hyaline layer occurs. So once the organic matrix is laid down, the next process is mineralization of that organic matrix. So the organic matrix begins within the hyaline layer. That is the initial center of mineralization. So from the hyaline layer, this is the Tom's granular layer, and from this hyaline layer, what happens is that the mineralization spreads towards the radicular, the rest of the radicular dentin, and the last part of the radicular dentin that is getting mineralized would be the hyaline layer. That is the outermost portion of the radicular dentin. And from once the hyaline layer gets mineralized, what will happen? It continues into the fibrous fringe secreted by the cementoblast. So here, this type of mineralization, that is the primary cementum, the mineralization of the primary cementum, it occurs very slowly and in a very linear way. So at this stage, the cementum can be classified as primary intrinsic fiber cementum. Why? Because the cementum that is laid down contains only the fibers secreted by the cementoblast. So it has only the intrinsic fibers. So at this stage, we call the cementum as primary intrinsic fiber cementum. Thus, the few layers of acellular cementum are attached to dentin. So, at the same time, this cementum doesn't have cementocytes. So, the primary cementum, initially the first formed primary cementum is acellular intrinsic fiber cementum. But in the later stages, the principal collagen fibers of the periodontal ligament become continuous with the fibrous fringe of cementum. And once the periodontal ligament fibers get attached to the fibrous fringe, this cementum is classified as acellular extrinsic fiber cementum. So I have already mentioned that as a radicular dentin is formed, some cells of the dental follicle gets differentiated to form cementoblast as well as fibroblast. And this cementoblast will secrete collagen fibers, and as well as the fibroblast also secretes collagen fibers. So these, when the primary cementum doesn't have cementocytes, So the primary cementum is acellular. It doesn't have cementosite, so it is acellular. So when the primary cementum initially, it has got only the intrinsic fibers, then you call it as primary intrinsic fiber cementum. And afterwards, in the later stages, this intrinsic fibers of the cementum is getting attached to the principal fibers of the periodontal ligament. So in that case, the fibrous fringe is getting attached to the extrinsic fibers that is secreted by the fibroblasts of the periodontal ligament. At this stage, you call the cementum as acellular extrinsic fiber cementum. So in permanent teeth, attachment occurs. That is, the extrinsic fiber is getting attached to the fibrous fringe of the cementum. That attachment occurs only after the tooth has erupted into the oral cavity. Thus, the acellular cementum lining the root before eruption may be considered as acellular intrinsic fiber cementum. Only after eruption, this periodontal ligament fibers, that is extrinsic fibers, get attached to the fibrous fringe, forming the acellular extrinsic fiber cementum. Now, just like enamel and dentin, cementum is also laid down periodically, and there are periods of rest and periods of activity. The periods of rest are associated with the incremental lines, and in cementum you call it as incremental lines of Salter. These lines are closer in acellular cementum as the cementum is formed very slowly. And so what are these incremental lines? They are hypermineralized area with less collagen fibers and more ground substance. Now, there is a type of cementum known as acellular afibrillar cementum. So from the term itself, you can know there is no cells, there is no fibrils. So how this type of cementum is formed? This is usually present at the coronal portion. And so this type of cementum, it is also known as the coronal cementum. So how this type of cementum is formed? You know that as the enamel is completely formed, it would be covered by the reduced enamel epithelium. So in cases where reduced enamel epithelium is damaged or lost, what happens? The newly formed enamel would get exposed to the dental follicular cells. And as a result, this induces the dental follicular cells to differentiate to form cementoblast. And this cementoblast secretes afibrillar matrix and then later calcifies. So this matrix is devoid of cells and collagen fibers, both extrinsic and intrinsic fibers. That is why it is known as acellular afibrillar cementum. And it has got a thickness of 1 to 15 micrometer. So that's about the primary cementum. Secondary cementum is that type of cementum that is formed after the primary cementum. So after the formation of acellular cementum or primary cementum, a less mineralized cementum is formed, and that is called the cellular cementum. So this type of cementum, as I've already mentioned, acellular cementum or primary cementum is present at the cervical 1/3 of the root, whereas secondary cementum is seen mainly in the middle and the apical third and also in the furcation areas. So unlike primary cementum is laid down very slowly, but secondary cementum, it's a rapid process. So as the matrix is laid down very rapidly, what happens is that some of the cementoblast gets entrapped within the organic matrix and cementocytes would be seen in the secondary cementum. So the cementoblast secrete the collagen fibers and the ground substance. And as the cementoblast secrete these fibers, you call it as intrinsic fibers. They run parallel to the root surface and doesn't extend into the periodontal ligament. At this stage, you call such type of secondary cementum as intrinsic cellular cementum because it has got entrapped cementocyte and also it contains collagen fibers that is secreted by the cementoblast. So this type of secondary cementum, you call it as cellular intrinsic cementum. So when you see the incremental lines in the cellular cementum, they are widely spaced because of the increased rate of formation, whereas in acellular, it is closely placed. Now, what is a cementoid tissue? Unmineralized pre-cementum present on the surface of cementum, you call it a cementoid. The cementoid tissue is lined by cementoblast. Cementoid tissue is not observed in the primary cementum or acellular cementum. Now, another type of cellular cementum is cellular mixed fiber cementum. Mixed fiber means it has got both intrinsic and the extrinsic fibers. Cellular mixed fiber cementum is a variant formed at a faster rate with less mineralized fibers. The collagen fibers are derived from the periodontal ligament fibroblast and also from the cementoblast. These intrinsic and extrinsic fibers form an intricate pattern running between each other at almost right angles and with different orientations. The next form of cellular cementum is cellular mixed stratified cementum. In this type of cementum, the cellular intrinsic fiber cementum alternates with acellular extrinsic fiber cementum. It is formed by cementoblast and fibroblast. It appears primarily on apical third of the root and furcation areas. The thickness varies from 100 to 1000 millimeter. Now, there is a layer known as intermediate cemental layer. Sometimes dentin is separated from cementum. Usually the cementum and the hyaline layer of dentin has a very strong union. But in some very rare cases, dentin is separated from cementum by a zone known as the intermediate cemental layer, which does not exhibit characteristic feature of either dentin or cementum. So, and this appears completely structureless. It is also known as the hyaline layer. This layer is predominantly seen in the apical two-thirds of the roots of molars and premolars. It is believed that this layer, that is the intermediate cemental layer, represents areas where cells of Herdwick's epithelial root sheath become trapped in a rapidly deposited dentin or cementum. The intermediate cementum layer is considered to be of dentinal origin. It contains but no tubules. It appears very similar to aprismatic enamel, as it is an amorphous layer of non-collagenous material devoid of odontoblast and cementoblast. The probable function might be to seal the sensitive root dentin. So this is the difference between acellular and cellular cementum. Acellular means no cementocytes. Cellular cementum, cementocytes present. Acellular deposition rate is slow. It's a first form layer found more at the cervical third of the tooth, also known as primary cementum. Sharpie's fibers are well mineralized. Incremental lines are regular and closely placed. Whereas in cellular cementum, cementocytes are present. Deposition rate is fast. Formed after cellular cementum, mainly seen at the apical and interradicular and also at the furcation areas, also known as secondary cementum. Incremental lines are irregular and placed wide apart. So differences between cementocytes and osteocyte. The main differences lie in the lacunae and the canaliculi. Lacunae of cementocyte are ovoid or tubular. Basically, these cells lie in a lacunae. So these lacunae is ovoid or tubular in the case of cementocyte, and in the case of osteocyte, the lacunae, the shape of the lacunae is oval. Canaliculi are less complicated and sparse and face towards a periodontal ligament for cementocyte, whereas osteocyte, the canaliculi are radiating, more dense and arranged in a complex network. Then immunohistochemistry, it is positive. Cementocyte is positive for fibromodulin and lumikin, and osteocyte, it is negative. Coming to the types of cemento-enamel junction, overlap type, there is a edge-to-edge type and there is a gap type. So overlap comprises of 60 to 65%, where the cementum overlaps the enamel. Edge to edge, it comprises of 30%, and in very rarely 5 to 10%, there would be a gap junction where the cementum and enamel doesn't meet. Coming to the functions of cementum, anchorage, adaptation, and repair. These are the main functions of cementum. Anchorage, it furnishes medium for the attachment of collagen fibers that bind the tooth to alveolar bone. Adaptation, deposition of cementum in the apical area can compensate for loss of tooth substance in an apical area, loss of tooth substance from occlusal wear. The continuous deposition of cementum is of considerable functional importance. Repair, damages to roots such as fractures and resorptions can be repaired by the deposition of new cementum. Cementum formed during repair resembles cellular cementum because it forms faster. A short note on hypercementosis. So what is hypercementosis? It is an excessive Deposition of cementum on the root surface. It can be diffuse or circumscribed. Etiology, it can be localized. If it is a localized hypercementosis, it can be found in teeth that are exposed to great stress. It could be associated with a tooth having chronic periapical inflammation. Any injuries to cementum can cause hypercementosis. Then generalized thickness of the cementum can be seen in neoplastic and non-neoplastic disease and certain metabolic diseases also. Now, what do you mean by excementosis? Excementosis, they are small knob-like localized projections observed in areas in which enamel drops have developed in the dentin. Radiographically, thickening and blunting of the root can be seen. Okay, this is a radiographic appearance of hypercementosis. Okay, the next topic is periodontal ligament. The periodontal ligament occupies the periodontal space, which is located between the cementum and the periodontal surface of the alveolar bone. So the periodontal ligament, it's a fibrous connective tissue that is noticeably cellular and contain numerous blood vessels. It comprises of cells as well as extracellular matrix consisting of fibers and ground substance. So the cells of the periodontal ligament involves synthetic cells, resorptive cells, progenitor cells, epithelial cell rest of Malassez, and defense cells. Synthetic cells include osteoblasts, fibroblasts, and cementoblasts. So coming to the synthetic cells, all synthetic cells share some unique characteristics. They all show a large open-face nuclei, vesicular nuclei containing prominent nucleoli. They tend to have abundant cytoplasm that tends to be hematoxophilic due to large number of rough endoplasmic reticulum covered by ribosomes, and they have large number of mitochondria. Usually all synthetic cells will have a large nuclei and large number of intracytoplasmic organelles. So coming to the first synthetic cell, that is osteoblasts. Osteoblasts are basically they are bone-forming cell. These cells are cuboidal in shape with a prominent round nucleus placed at the basal end of the cells. The cells appear basophilic due to the presence of abundant rough endoplasmic reticulum. So these are the osteoblasts. The cells contact one another through desmosomes and tight junctions. Osteoblasts are also in contact with underlying osteocytes through cytoplasmic process. Coming to the next synthetic cell, that is a fibroblast. They are the predominant cells in the periodontal ligament. These fibroblasts are regularly distributed throughout the ligament and are oriented with their long axis parallel to the direction of the collagen fibrils. The fibroblasts near cementum are derived from ectomesenchymal cells of the investing layer of dental papilla, while fibroblasts near alveolar bone are derived from the perivascular mesenchyme. So these fibroblasts, they are large cells with extensive cytoplasm and abundant organelles associated with protein synthesis and secretion. The nucleus occupies a large volume of the cell and contains one or more prominent nucleoli. The fibroblasts show extensive network of all these intracytoplasmic organelles like Golgi apparatus, rough endoplasmic reticulum, abundant secretory granules containing type 1 collagen molecules. The cells have well-developed cytoskeleton and show contacts of adherents and gap junction types. The fibroblasts of periodontal ligament have cilia. The cilium is different from those in other cell type as it has only 9 tubule doublets. The role of the fibroblast is to produce the structural connective tissue, proteins, collagens, and elastin, as well as ground substance, proteoglycans, glycoproteins, and glycosaminoglycans. They have dual function. They are responsible for the formation and also remodeling of the periodontal ligament fibers and a signaling system to maintain the width of the periodontal ligament. They have the ability to both synthesize and shape the proteins of the extracellular matrix in which collagen fibrils form bundles. Coming to the next synthetic cell, that is cementoblasts. These cells line the surface of cementum and they form the cementum. They are almost cuboidal with a large vesicular nucleus with one or more nucleoli and abundant cytoplasm. All the organelles required for protein synthesis and secretion are present. And cells actively depositing cellular cementum exhibit abundant basophilic cytoplasm and cytoplasmic processes. while cell-depositing acellular cementum do not have prominent cytoplasmic processes. And just like other cells, this also shows gap junctions and desmosomes and has receptors for growth hormone and epidermal growth factor. Now coming to the resorptive cells, we have osteoclast, fibroblast, and cementoclast. So osteoclasts, they are cells that resorb bone and tend to be large and multinucleated. The characteristic multinucleated cells usually show an eosinophilic cytoplasm, that is a pink color cytoplasm. When viewed in light microscope, the cells may sometimes appear to occur base in bone. They usually, wherever resorption is occurring, these multinucleated osteoclasts, they lie on the surface of the bone to be resorbed. And what happens? So they usually rest on these resting base, and they are known as Hauschup's lacunae. So once they come and rest on the particular bone surface to be resorbed or on the Hauschup's lacunae, the next thing is they form characteristic fold. The plasma membrane shows characteristic folding, and this region is known as a ruffled or the striated border. So what is the significance of this ruffled border is that it separates or it seals the bone to be resorbed from the rest of the bone. That is the purpose of this ruffled border. So this ruffled border is separated from the rest of the plasma membrane, where the cytoplasm is devoid of all the organelles, and this is known as the clear zone. Clear zone is the plasma membrane, that is the ruffled border plasma membrane that is closely applied to the bone, is separated from the rest of the plasma membrane through a clear zone. So the area of the bone that is sealed off by the ruffled border is exposed to a highly acidic pH by virtue of the active pumping of protons by the osteoclast into this environment. And the bone related to the ruffled border undergoes resorption. So that is about osteoclasts. Fibroblasts, as I've already mentioned, they have a dual function, that is both remodeling, resorption, and secretion. The third one is the cementoclast. Cementoclasts resemble osteoclasts and are occasionally found in normal functioning periodontal ligament. And unlike bone, cementum is not resorbed. Bone is actually remodeling or getting resorbed in normally, that is a physiological condition. But in cementum doesn't resorb like that. It undergoes only continual deposition during life. Resorption occurs only under certain circumstances in cementum, and these instances, cementoclasts often located in Hauschup's lacunae, are found on the surface of cementum. Coming to the progenitor cells. Progenitor cells are basically, they are the cells that have the capacity to undergo mitotic division. And these cells, when needed, they can differentiate into various types of cells. And these cells appear to be in highest concentration in locations adjacent to blood vessels. And they all exhibit some of the classical cytological feature of stem cells, including small size, close-phase nucleus, and very little cytoplasm. They replace differentiated cells dying at the end of their lifespan or as a result of trauma. They are progenitor cells. Epithelial cell rest of Malassez, they are the remnants of the Herdwick's epithelial root sheath cells. These cells were first described by Malassez in 1884. At the time of cementum formation, the continuous layer of epithelium that covers the surface of the newly formed dentin breaks into lace-like strands. So, the distribution of these cells varies according to site and age. They are less numerous in older individuals and more numerous in children. Up to the second decade, these cells are most commonly found in the apical region. Later, they are mainly located cervically in the gingiva above the alveolar crest. These cells may proliferate to form cysts and tumors, and also it can undergo calcifications to become cementicles. And last comes the defense cells, that includes mast cells, macrophages, and eosinophils. All these are hematopoietic stem cell. Mast cells, they are round or oval shaped cell, and it has got numerous cytoplasmic granules containing histamine and heparin. So the main function is to the release of histamine in the extracellular environment, causing proliferation of endothelial cells and mesenchymal cells. Macrophages, they are horseshoe-shaped or kidney-shaped cell with dense and uneven layer of peripheral chromatin. They have a dual role, that is phagocytose the dead cells, and also it secretes growth factors that regulate the proliferation of fibroblasts. Eosinophils, they are bilobed nucleated cell, and they have got numerous granules in the cytoplasm having bright red staining properties with acidic dyes such as eosin. The main function is again phagocytosis. So that finishes the cellular compartment of the periodontal ligament. Now we move on to the fibers. So the connective tissue fibers are mainly collagenous, and they may be small amounts of oxytocin and reticulin fibers. Collagen is a protein composed of different amino acids. The collagen is gathered. Small collagen fibrils gathered to form bundles, and these bundles of the periodontal ligament are known as the principal fibers. The main types of collagen in the periodontal ligament are type 1 and type 3. More than 70% of periodontal ligament is type 1 and 20% is type 3. Small amounts of type 5 and type 6 collagens and traces of type 4 and type 7 collagen are also found in the ligament. So the principal fibers of the periodontal ligament can be grouped under 5 categories: alveolar crest group, horizontal group, oblique group, apical group, and interradicular group. Alveolar crest group, from the name itself, we can understand that it extends obliquely from cementum over the alveolar crest of fibrous layer of periosteum covering alveolar crest, and also is extended obliquely from cementum just beneath junctional epithelium to the alveolar crest. That is, it extends from the alveolar crest of the alveolar bone to the cementum. So the main function is to resist tilting, intrusive, extrusive, and rotational forces. Horizontal groove, it extends at right angles to the long axis of the tooth from cementum to the alveolar bone. These are the horizontal groove. They are parallel to the occlusal plane. They are inserted as Sharpie fibers. Sharpie's fibers are embedded portion of the principal fibers of the periodontal ligament. It is seen both in cementum as well as in alveolar bone. So this horizontal groove, the main function is to resist horizontal and tipping forces. Oblique groove, it extends into alveolar bone coronal to the attachment to cementum. These are the oblique groove. Resists vertical and intrusive forces and is the most numerous of the periodontal ligament fibers. Apical groove, these are the apical groove. They are not seen in incompletely formed roots. They extend from the root tip and radiate through the periodontal space into fundus of the bony socket. This one. It resists luxation, prevent tooth tipping, protect blood vessels and nerves, and traverse through the periodontal ligament space at the root apex. And the last one is the interradicular groove. It extends from cementum into cementum from the crest of the interradicular septum of multi-rooted teeth. This is the interradicular groove. It resists tooth tipping, torque, and luxation. Sharpie's fibers, collagen fibers that are embedded into cementum on one side of the periodontal space and into alveolar bone on the other. These embedded fibers are known as Sharpie fibers. They are more numerous but smaller at the attachment to cementum than alveolar bone. Once embedded in either the wall of the alveolus or cementum, Sharpie's fibers calcify to a certain degree. Sharpie's fibers in primary acellular cementum are mineralized fully. Those in cellular cementum and bone are mineralized only partially at their periphery. Now, what is intermediate plexus? It was thought that the principal fibers frequently followed a wavy course from the cementum to bone and are joined in the mid-region of the periodontal space, giving rise to a zone of distinct appearance, the so-called the intermediate plexus. And the plexus was also considered to be an area of high metabolic activity. But research over the past few years demonstrated that once cemental fibers meet and fuse with the osseous fibers, no such plexus remains. Secondly, the entire periodontal ligament is metabolically active and not just the middle or the intermediate zone. Studies also indicate that areas of highest activity are fiber terminals near cementum and bone and not in the middle. So the latest concept is that the intermediate plexus is just artifactual. It doesn't exist. Now, a short note on oxytocin fibers. They are basically immature elastic fibers. These fibers are approximately 0.5 to 2.5 micrometer in diameter and appear to have elastin and type 4 collagen. The orientation of the oxytocin fibers is quite different from that of the collagen fibers. They run in an axial direction, one end being embedded in cementum or possibly bone, and the other often in the wall of a blood vessel. In the cervical region, they follow the course of gingival and transeptal fibers. Within the periodontal ligament proper, these fibers are longitudinally oriented, crossing the oblique fibers perpendicularly. The function is supporting blood vessels of the periodontal ligament. The main function of the oxytocin fibers is they support blood vessels of the periodontal ligament. And coming to the functions of the periodontal ligament, that is supportive, sensory, nutritive, homeostatic, and eruptive. So supportive, when a tooth is moved in its socket as a result of forces acting on it during mastication or through application of orthodontic force, part of the periodontal space would be narrowed and the periodontal ligament contained in this area would be compressed. So other part of the periodontal space where the force is not applied will be widened. So this compressed periodontal ligament provides support for the loaded teeth. And the pressure in the numerous vessels in that compressed periodontal ligament provides a hydraulic cushion for the support of the teeth. So that is how it supports the tooth. Sensory, periodontal ligament through its nerve supply provides a most efficient proprioceptive mechanism, allowing to detect the application of the most delicate forces to the teeth. This protects both supporting structures of the tooth and the substance of the crown from excessive masticatory forces. That is about sensory. Nutritive, the ligament contains blood vessels which provides anabolites and other nutrients required for the cells of the ligament. The blood vessels are also concerned with removal of catabolites, that is the waste products. The fourth function is homeostatic. The cells of the periodontal ligament have the capacity to resorb and synthesize the extracellular substance of the connective tissue of the ligament. It is happening throughout the life of the tooth, thus it's maintaining the balance. All these are exquisitely controlled by various mechanisms. The preservation of the periodontal ligament width throughout the mammalian lifetime is also an important measure of periodontal ligament homeostasis. And the last function is it helps in eruption. Now, the last topic is alveolar bone. Bone is a living tissue which makes up the body skeleton and is one of the hardest structures of the animal body. Histologically, bones can be classified as mature bone and immature bone, and mature bone is further classified as compact and cancellous bone. All mature bones have a dense outer sheet of compact bone and a central medullary cavity. The medullary cavity is filled with red or yellow bone marrow in living bone. This marrow cavity shows a network of bony trabeculae. So if you take a longitudinal section of a bone, what we can see is that the outermost portion, this is the compact bone. Inside you have a medullary cavity, and the medullary cavity shows thin plates, so bars of bone deposition, and these are basically known as trabeculae. This is the characteristic feature of a spongy bone. So coming to the compact bone is otherwise known as cortical bone. It consists of tightly packed osteons or Haversian system forming a solid mass. Since the bone is arranged in layers, it is also called as lamellar bone. So if you take a compact bone, the outer aspect of the compact bone is surrounded by condensed fibrocollagen layer, and it is known as a periosteum. This periosteum has got two layers, an outer layer which is dense and irregular connective tissue. It's known as the fibrous layer of the periosteum, and an inner osteogenic layer or cellular layer that is forming the inner layer of the periosteum. Now, if you take, so just inside the periosteum, if you see, the inner surface of compact bone is covered by a thin cellular layer called the endosteum. So this is the, if you take a compact bone, if you take the compact bone, the outermost portion is lined by periosteum, and the inside, that is the medullary cavity, where it is facing the medullary cavity, the inside layer, you call it as endosteum. And both at the periosteal surface and at the periosteal surface and at the endosteal surface, the lamellae, the bone is arranged in parallel layers. The bone is deposited in parallel layers, and these are known as the circumferential lamellae. So the outermost covering is the periosteum, innermost lining the medullary cavity is endosteum, and at the periosteal and adjacent to the endosteal surface, the bone is laid down in the form of parallelly arranged lamellae, and these lamellae is known as circumferential lamellae. Deep to the circumferential lamellae, the lamellae arranged as small concentric layers. So deeper to the circumferential lamellae, you see the bony matrix is laying down or laid down as concentric layers or circles around the central vascular or Haversian canal that contains blood vessels. So Haversian canals and the concentric lamellae, the central blood vessel and the surrounding concentric lamellae together known as the osteono-Haversian system. And these adjacent Haversian systems are interconnected by Walkman's canals, that is channels that contain blood vessels, creating a rich vascular network throughout the compact bone. So that is the histology of compact bone, Haversian system or osteon. Now coming to the structure of the alveolar bone. As a result of its adaptation to function, two parts of the alveolar process can be distinguished. One is the alveolar bone proper, and the other part is the supporting alveolar bone. Alveolar bone proper is that portion of the bone that is covering the roots, or that is coming closer to the cementum of the tooth. So this alveolar bone proper can again be divided into lamellated and bundle bone. It surrounds the root of the tooth and it gives attachment to principal fibers of the periodontal ligament. Alveolar bone proper is the one that surrounds the root of the tooth and it gives attachment to principal fibers of the periodontal ligament. The lamellated bone, it contains osteons. The lamellated bone as well as the bundle bone, both are compact bone. So histologically, when you see a compact bone, we can see the same histological structure as I've explained before. A circumferential lamellae, you have a concentric lamellae around a Haversian vessel, and you have a Walkman's canal. So both the lamellated and the bundle bone, they are compact bone. Bundle bone is that bone in which the principal fibers of the periodontal ligament are anchored. The term bundle was chosen because the bundles of the principal fiber continue into the bone as Sharpie fibers. So this is the alveolar bone proper. And this is the supporting alveolar bone. It contains of cortical plates and the spongy bone in between the cortical plates. They form the supporting structure. So the bundle bones, they contain fewer fibrils and lamellated bone, and radiographically, this bundle bone can be seen as a radio-opaque line around the root of the tooth. This arrowhead is pointed towards the radio-opaque line, and that is the lamina dura. So radiographically, the bundle bone is referred to as lamina dura because of the increased radio-opacity, which is due to the presence of thick bone without trabeculations. Alveolar bone proper, which forms the inner wall of the socket, is perforated by many openings that carry the branches of the interalveolar nerves and blood vessels into the periodontal ligament, and is therefore also known as the cribriform plate. Now, what is the Zucker-Candle and Hirschfeld canals? They are basically the interdental and interradicular septa contain the perforating canals of Zucker-Candle and Hirschfeld. They are basically nutrient canals. which houses the interdental and interradicular arteries, veins, lymph vessels, and nerves. And last is about the bone remodeling. So bone remodeling, what do you mean by the term bone remodeling? Bone remodeling is a continuous physiological process that occurs in adult skeleton in which bone resorption is followed by bone formation. So bone remodeling is performed by clusters of bone resorbing osteoclast and bone forming osteoblast. So this bone remodeling can be divided into 4 stages. First is the resting phase or the quiescent phase, followed by a resorptive phase, followed by a reversal phase, followed by the osteogenesis or formation of osteoblast, formation of bone, and then again resting phase. So resting phase is that no resorption, neither resorption nor deposition occurs. So after the resting phase, what happens is that the next is the resorptive phase. So osteoblasts respond to signals generated by osteocytes and they recruit osteoclast precursors at the remodeling site. Remodeling means resorptive site. So once they get, once they receive signal from the osteoblast, the osteoclast precursor cell, they either can be a macrophage or a monocyte lineage, they differentiate to form multinucleated giant cells known as osteoclasts. And these osteoclasts will be located in house-shaped lacunae. And I have explained before that they form the ruffle border and they undergo resorption of that particular area of the bone. And so resorption occurs. And at the end of this resorption process is marked by a cemental line that is strongly basophilic. Okay, this marks the end of bone resorption prior to the bone formation. This is known as a reversal phase because at the end of the resorption, bone resorption, at the end of the bone resorption, what is happening? There is a formation of a cemental line. Cemental line, and this line is known as the reversal line. Reversal line, it appears to be highly hematoxyphilic and highly irregular. So in the reversal phase, what happens is that there would be differentiation of the osteoblastic precursors. So resorption has ended. And now when it reaches the reversal stage, what happens? The osteoblasts are getting differentiated in the reversal phase. And this leads to the next stage, that is formation. So in bone formation stage, activated osteoblasts, they lay down new bone material until the reserve bone is entirely replaced by this new one. So during this phase, there would be periods of rest. It's just like any other mineralized tissue. This bone is deposited periodically. And so between each session, there would be periods of rest. And this line that is separating the periods of formation, you call it as a resting line. And this resting line compared to the reversal line is more regular in appearance. And once the bone is formed, the formed bone completely replaces the already resorbed bone. Once a new bone is formed, then what happens? This enters into a quiescent phase again. And this thing continues. So in the quiescent phase, the bone would be lined by flattened cells, that is an osteoblast on the bone surface, until a new remodeling cycle is triggered. So this is the whole process of bone remodeling. And this is completely a physiologic process. It occurs naturally. And so these are the resting lines. These are the resting lines. Resting lines are seen between the bone formation. And reversal line is what? It indicates that, reversal line indicates that bone resorption is ended and next would be the new bone formation. That is the reversal line indicates. Resting line, these are the lines that is present between the formation or osteogenesis. So that finishes my presentation. Thank you all for your patient listening. And my heartfelt gratitude to Deepu sir and Team Oral Pathology Anu Dental College for giving me this wonderful opportunity. Thank you all.